guys, welcome to Biomix Sub. In this today's video, we are going to discuss about DNA replication. So, what do you mean by DNA replication? Or we can tell that what do you mean by only replication? Replication means a copy of something. So, in this case, DNA is replicating. That means the copy of DNA is taking place. So, our DNA which is present inside the nucleus, it is of double stranded. Like we can see here that it is double stranded. It is having one 5' end, it is having one 5' end, as well as it is having a 3' end. So in this way, these two are anti-parallel with each other. It is having a positive strand and it is also having a negative strand. So this blue colored DNA strand which you are seeing, that is the parent DNA. It is the parent DNA. So there are certain enzymes which takes place and copies this parent DNA, and they are exact copy. So what is there in case of a DNA? There are purine as well as pyrimidine. We can see that the adenine which is present, it is forming bond with thymine and two hydrogen bonds are formed between them. And when cytosine base pair with guanine, it is having 3 hydrogen bond. Okay, these are two things which are taking place in this DNA replication. So there are certain enzymes which are taking place in the DNA replication. They are the DNA polymerase, helicase, single-stranded binding protein. So before going to that topic, we will discuss where this DNA replication is taking place. So this DNA replication which is present, it is done in both prokaryote cell as well as eukaryotes. In case of bacteria, which is a prokaryote, the DNA replication takes place in the cytoplasm or cytosol. It is taking place in the cytosol. The process of DNA replication, transcription as well as translation takes place in the cytosol in case of prokaryotes. But what happens in case of a eukaryotes? The process of DNA replication and transcription, it takes place inside the nucleus, and after the mRNA is formed, after some modification, it is being transported into the cytosol for the synthesis of protein. So we will not discuss now about the protein or mRNA. We will discuss how the basics of DNA replication, what are the things required for DNA replication, what are the enzymes involved in this DNA replication. In the next video, we will discuss about one of the enzyme which is known as DNA polymerase. It is of many different types, like DNA polymerase 1 to 5 are present, and in the upcoming videos we will discuss separately about the prokaryotic and eukaryotic DNA replication. So let us see what are the things required for the process of DNA replication. When the DNA is forming its duplicate or copy of itself, what are the things necessary? Already we have discussed that the DNA which is present, it is of double stranded. It is a double stranded. So during the process of replication, this double stranded should be converted into single stranded. We can see here it requires, for the process of DNA replication, it requires a single stranded template. The other thing is it requires a primer. And this primer is synthesized by an enzyme which is known as primase. Okay, so this primase which is present, it is a DNA dependent RNA polymerase. Okay, here there is no involvement of DNA polymerase during the synthesis of primase. Why? Because if there is no free 3' OH group, the DNA polymerase cannot help in binding the nucleotides, or it cannot form the copy. But in case of RNA polymerase, it can form a short strand of nucleotide. Okay, it will in case of RNA polymerase, it can form a short strand of nucleotide by seeing the DNA. So it is dependent upon the DNA by which it can synthesize a fragment. Okay, so now it has given 3' OH group. So during this time, what happens? Another polymerase will come, that is known as DNA polymerase, and it will help in the extension. Okay, now let us see how this process is taking place. So we are seeing here that there is a DNA template which is of single stranded. This is a single stranded DNA template, and that is a primer. It is synthesized. This primer is having free 3' OH group. Okay, so there are upcoming nucleotides. These nucleotides which are coming, it is having 3 phosphate group like alpha phosphate, beta phosphate and gamma phosphate. The 3' OH group which is present, it makes a attack with this alpha phosphate of the upcoming nucleotide, and it forms a bond which is known as phosphodiester bond. It is known as phosphodiester bond. In this way, the replication takes place, and during the formation of bond, there is release of the phosphate. 2 phosphates are released, that is beta and gamma phosphate are released. They are known as pyrophosphate. phosphate. Okay, so the next thing which we now want to need to know is replication fork. So from what do you mean by replication fork? There is a specific site from where this DNA replication can take place. So this DNA replication which is present, in most cases it is a bidirectional type. That means it is starting from a point and in two direction it will show the DNA replication. So one strand is known as leading strand, the other strand is known as lagging strand. Now let us see what is leading strand, what is lagging strand. When there is only one primer is formed here by the involvement of the primase and there is continuous synthesis of this strand, it is known as leading strand. But if multiple primers are present here for the synthesis of the strand, it is known as lagging strand. So what happens here, that is in this DNA we can see that this is a 5' end and this is also the 5'. This is not the 5' end, this is the 3' end if we see the total. But as this DNA replication, origin of replication is starting from here. So from here it is 5' end and here is 3' end. Okay, so from here it is forming a continuous synthesis is taking place. It is known as leading strand. But in this case we can see when this fork is opening, it is opening a little bit. So from here the synthesis starts. Okay, because this is, in this case it is a 5' end. So from 5' end the synthesis will not start. From the 3' end of the parent strand the synthesis will start. So from here the primer is formed and there is continuous synthesis. Okay, in this way this is the first part where the DNA is synthesized is DNA, where the DNA has replicated its parent strand into the daughter strand. When the fork is opening a little bit more, there is some other space which need to be replicated. So there is another one primase enzyme coming and synthesizing a primer. Here there is another primer which is synthesized and here the extension takes place. In this way multiple strands are synthesized here, then they are joined together. This continuous strand, the strand which is formed continuously, it is known as leading strand. If there are multiple breaks, they are known as lagging strand. In this way, this is the leading strand because it is starting from 5' end and it is ending in 3' end. But in this case, it is the lagging strand. Okay, so there will be a question from which direction the DNA replication is taking place. The DNA replication takes place in 5' to 3' direction. Okay, it takes place in 5' to direction, but this is of the new strand. If it is taking place in the 5' to 3' direction, what will be the sequence or what will be the direction of the parent strand? It will be 3' to 5' of parent strand. Okay, so now next we will see what are the enzymes required for the whole process to be done. So the first enzyme is helicase. What is this helicase? This helicase is a hexameric protein. And it requires ATP for its work to be done. So what is the work of this hexameric protein or the helicase? As we already discussed that the DNA for its replication, it requires a single stranded. When it is in the double stranded form, it will not replicate. So the work of helicase enzyme is, it will help in the formation of single stranded of the DNA by unwinding the DNA. Okay, like we can see here that this is in the helical form. This helicase is coming and binding in this region. By the use of ATP, it is helping in removing the strand into single strand, and when it helps in the formation of single strand, that is positive supercoil in the back. In this, suppose here the DNA is making the single stranded over here, but when the helicase moves forward, what it does, it helps in positive supercoiling. Okay, so next here we can also see that the same thing here, the helicase enzyme is coming and binding. There is requirement of ATP and the double stranded form is converted into single stranded form. Okay, so next is the single stranded binding protein. Here there is another one enzyme which is known as single stranded binding protein. Okay, so the helicase has done its work, like it is formed, it has given a single stranded. so the single stranded can again recoil. Okay, so for preventing this recoiling of the single stranded, there is another one protein which is known as single stranded binding protein. This protein binds in the single strand of this DNA. If this is a DNA which is of single stranded, it binds in this region for preventing it from again recoiling. Okay. The next enzyme is the topoisomerase. What is this topoisomerase? It is another one enzyme which is required during the DNA replication. This topoisomerase is again of two types, like topoisomerase 1 and topoisomerase 2. So when the DNA helicase does its work, this topoisomerase is required. When the DNA helicase does its work, it helps in positive supercoiling. So this positive supercoiling needs to be removed. It is being removed by this topoisomerase by breaking this DNA. Okay, this DNA is being cut. This topoisomerase cuts the single-stranded DNA. It cuts the single-stranded DNA, and the topoisomerase 2 cuts the double-stranded DNA. It cuts the double-strand DNA. Okay, after the cutting of the strand, it again twists itself to form the linear form of the DNA. Again it rejoins. Due to which the positive supercoil will not form, it will be like a simple DNA. Okay, this is the work of topoisomerase. The next enzyme which we will discuss is the DNA polymerase, which will be discussed in the other video. This DNA polymerase is helping. These are the three enzymes which are required during the DNA replication. The main enzyme which is required during DNA replication is DNA polymerase, which help in the replication or bringing the copy of the parent strand. So there are different type of DNA polymerases, like DNA polymerase 1, 2, 3, 4 and 5. There are different function of each. So this will be very last video, so we will discuss that in next video. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.